Molecular science. Today we'll discuss an interesting topic called as exosomes. We'll be talking some important facts about exosomes. Exosomes are lipid vesicles which shuttles proteins and genetic information between both neighboring and distant cells. Now, these extracellular vesicles has a potential to identify unknown cellular and molecular mechanisms in intercellular communications and in organ hemostasis and disease. They are just ubiquitous facts in our biological system. They were discovered in 1970s by Ross Johnstone, who identified the presence of small vesicles associated with transferrin. I will see what are exosomes. These are nano-sized extracellular vesicles measured about 50 to 150 nanometer. They are released from all cell types into the extracellular space. Exosomes are found in all biological fluids including blood, urine, cerebrospinal fluid, amniotic fluid, breast milk, seminal fluid, saliva and malignant effusion. Exosomes are endocytic. They carry valuable information as they carry RNA, mRNA, miRNA, that is microRNA, long non-coding RNA, DNA, proteins, lipids, metabolites from the cells in which they originate and from which they are shed. Also, their contents have been implicated in the development of drug resistance, cancer progression, and metastasis. And now we'll see about the functions of exosomes. When exosomes were initially discovered, they were thought to be garbage disposals. They were thought to eliminate unwanted cellular components, redundant or misfolded proteins, molecules, and metabolic waste from the cells. Later, it was discovered that exosomes have important physiological functions like cell-to-cell communications, immune response modulations, stem cells maintenance, and tissue repair. Along with these functions, exosomes also implicated in the pathogenesis of infections, neurodegenerative diseases, and cancers due to their ability to spread pathogenic agents and toxic neurodegenerative proteins aggregates, activate pro-tuberogenic pathway in cancers. Now let us see the content of exosomes. Now exosomes are rich with tetraspanins. Like here in this picture we can see the exosomes, like we can see the presence of CD9, CD63, CD81 which are specific to exosome. Exosomes also contain like integrin which are involved in cell adhesions, immunomodulators, antigen presentation, that is MHCs, MVB, exosomes biogenesis, tetraspanins, cholesterols, follitin, membrane transport, and transmembrane proteins. Along with this, exosomes also contain proteins like a cytoskeletal heat shock protein, nuclear. We have signal transducers, apoptosis proteins, RNA like mRNA, microRNA, pre-microRNA, yRNA, circular RNA, mitochondrial RNA, tRNA, and lots more. Small nuclear RNA, piRNA, genetic materials like DNA, double-stranded DNA, single-stranded DNA, and also viral DNA. Now this we can say the presence of viral DNA during the viral infection. So like exosomes can act as a transport medium of the viral DNA from infected cell to the neighboring or a distant cells. It is also rich in amino acids and certain metabolites which are involved in regulating the metabolic pathways. Now let's discuss about the heterogeneity of exosomes. Heterogeneity of exosomes means the exosomes they vary in their size, the content, their functionality as well as the source of the origin. Now, if we talk about the heterogeneity of exosomes in terms of size, now the size heterogeneity is due to the uneven imagination of the limiting membrane of multivesicular bodies. That is from where they originated. It is that uneven imagination of the membrane can lead to formation of different size of exosomes. Now this different size of exosomes, like here we can see the example, like exo1 has a size of 40 to 75 nanometer, exo-B is from 75 to 100 nanometer, and exo-C is from 100 to 160 nanometers. Now, coming to the content heterogeneity, here we have seen that some of the exosomes which are released, like exo1, they are rich in CD9, tetraspanin, nucleic acids of different combination of nucleic acid, protein X. Exosome 2, which may contain CD63, nucleic acids again, and protein Y. Exo3, which can contain like tetraspanin CD81, nucleic acids, and protein Z. Along with this, like we have now the the functionality. Now, based on the composition, the content of this exosomes, the functionality also varies. Like exo alpha, it plays a role in, or it imparts pro-survival signaling on the recipient cells. Exo beta, it provides a pro-apoptotic signals to the recipient cell. Exo gamma, it imparts immunomodulation to the recipient cells. Now, the size heterogeneity can result in different amount of exosomal content. Also, microenvironment and the inherent biology of cell can influence the exosomal content and their biomarkers, which in turn influence their function to the neighboring or the distant cell where they are exported. Now, here the next one is we'll think about source. Now the source also has important role, as like exo1, it is derived from brain, will have a different composition on heterogeneity. From pancreas will again have a different content and the functionality, as well as if it is derived from liver, it will have some different functionality. Now here we'll see the multi-parameter heterogeneity. In multi-parameter of heterogeneity, the first combination we can see, that is different combinations we can find here, like exo A1, B1, C1 of different composition, or you can say the composition that increases the complexity with the heterogeneity. So the total overall, the exosome complexity is very high if we talk in terms of heterogeneity which are released from a cell. Now the hallmarks of exosomes. That is, we will see the functions of exosomes. So exosomes has been implicated to have a role in regulation of transcription and translation. Then we have a role in a survival, proliferation, and apoptosis. As we have seen, the exosome content, they are rich with certain proteins, like some of the proteins impart proliferation, cell proliferation, some helps in the cell survival, and some may make the cell to enter into apoptosis. Next one is angiogenesis and wound healing. The next one is reproduction and development. In reproduction and development, the seminal fluids contain exosomes which are rich in microRNA. These microRNA are believed to increase the immunity of the human reproductive, local immunity of the human reproductive system, and to some extent it prevents the spread of HIV infection. Some reports are available. Based on the composition, they got it from microRNA from exosomes. That is, the next one is waste management. Exosomes are they have a role in waste management, as it was initially stated that exosomes are garbage disposals. Then they do play a role in host microbe interactions. And there is a role in metabolic reprogramming and regulation, especially with the like few example we can say is the regulation of cardiovascular diseases, neurodegenerative diseases, the other metabolic changes in the metabolites, which can which can lead to certain disease condition. Also in cellular differentiation and neoplasia, cellular migration and metastatics, and they do act as immune modulators. Also that I would like to discuss about that exosomes do contain some important proteins like TSG101, which is a tumor susceptibility gene 101, Alex, that is apoptosis link gene 2 interacting protein X, syndican 1, ESCRT, that is endosomal shorting complexes required for transport, proteins, phospholipids, tetraspanins, ceramides, sphingomyelinase, and SNARE. Now, these complexes and proteins, it depends upon the origin and during the biogenesis and the content of the exosome and the content which is being present in the exosomes. So this is briefly I have discussed about the what are exosomes, what are the functions of exosomes, the heterogeneity of the exosomes, and hallmark of exosomes. In the coming video, I'll be discussing about the biogenesis of exosomes. So hope you like this video and do subscribe my channel, and thank you for watching.